All right, so steps for contracting striated muscle. 1, you have to have some stimulus from the outside. And that stimulus is going to depolarize the cell membrane of the imaginary striated muscle cell that I'm trying to convince to contract. And since we're talking about striated muscle, we know that all striated muscle, regardless of its location, has some common features. And so step number 3 is so it spreads away and it goes towards a T-tubule. And, you know, in typical striated muscle, there's lots of T-tubules around. So once it's in the T-tubule, that directly causes so I'm going to put a superscript A here just to differentiate it. So that's the first thing it does. And it can also do thing B in some cases, which is it can also cause the opening of calcium channels in the membrane. But I have to stress, and this is what it sounds like the issue might have been. So now we can comfortably sort of circle back to cardiac muscle cell, because the way that step 5 happens, this is the major difference between cardiac muscle cell and skeletal muscle. Skeletal muscle relies mostly on A. Cardiac muscle relies on both A and B. So the reason I stopped you earlier is because you said something along the lines of, oh, I thought the calcium was causing the depolarization. They are two separate events. So this membrane depolarization that I'm discussing for cardiac muscle cell, the stimulus, so all the pink stuff is cardiac muscle. The stimulus for cardiac muscle is the SA node firing. I'm talking about a cardiac muscle cell that's probably in the atrium somewhere. So the stimulus is the SA node firing. That causes adjacent contractile cells to depolarize. This depolarization initially is sodium-based. So sodium influx, just like an action potential anywhere else. This sodium-based action potential causes the traveling wave of depolarization. Depolarization heads down T-tubule. So this is where we get into that part of the depolarization wave is that sodium influx in the contractile cell potential.
So which muscle cell or which muscle cell is depolarizing? Is it the contracting cell or the pacemaker cells in the step?
No, in step number 2, the pacemaker cell has already gone because pacemaker cells are part of the nodal system.
Okay, so then we're getting into the contractile depolarization?
Correct.
Okay.
The stimulus for the contractile cell to depolarize directly comes from the pacemaker cells depolarizing. So that calcium piece you were talking about that you were confused happens after the sodium as a result of having been stimulated. So now we have slow calcium channels. And then potassium. So the step 2 initial depolarization is not calcium based. That happens later. So at the outset, initially, the contractile cell potential, which is what this is, this part of it looks just like any other action potential. So we get a steep depolarization up past 0 to positive 20 or 30 due to sodium ions entering the cell. The only part that gets weird or funky or departs from what we know about action potentials is the next step. Does that make a little bit more sense now?
Yeah, that helps because then it ties in the 3 basic steps into these steps.
Yeah, so the thing I think a lot of students miss, and this is an issue of relating structure to function, is I show you 2 different, oopsie, that's all I wanted to do. Thank goodness for the undo button. So pink is still cardiac muscle, but I show you 2 different charts. One looks like the contractile cell potential, which I just drew, and then the other one looks like this, which is the pacemaker potential. And the thing that students miss is how does this cause that? So this depolarization, let me get another color. Let's do orange. No, not orange. I used that already. How about purple, magenta? So this depolarization here, this is the stimulus that starts that.
And this pacemaker potential, that's still tied into that first step.
Correct.
And then once you get into the second step, then you're talking about, so you get your stimulus, the SA node's firing, you're having the pacemaker potential happen. Once that happens, then the pacemaker potential now sends its information down to depolarize the contractile cells.
Yes. So there's a very, very small difference in time between those two things. It's only a millisecond or so. The reason, so think about what this means right here. If I see this line going up, what that means is sodium is entering the pacemaker cell, right? Because all these charts are just lines that represent ion movement. So when the pacemaker potential's line is increasing, that means sodium ions are entering the pacemaker cell. The reason I've drawn this connecting bubble between that leg of the pacemaker potential and this spot where we're getting the contractile cell up to threshold is because, well, let me ask you guys this. Remember when you had to look at cardiac muscle in 160 and in 241? There was a specific feature of cardiac muscle that you had to identify and recognize. Anybody remember what that is?
Are you talking about in terms of the binding site for this, or?
No, I'm talking about when you look at a microscope slide of cardiac muscle, there's little thingies that you have to find, and they're hard to find.
Was it striations?
Nope. Striations are a result of the interlocking sarcomeres. There's another thing that are specific to cardiac muscle. Intercalated discs ringing a bell?
Yeah.
Yeah, so this is one of the reasons why A and P is so tricky, is because stuff like that falls out of your brain, but you actually really need it for later. So intercalated discs are an annoying thing to find on a microscope, but they really matter. Here's why. Intercalated discs are cell junctions that join two cardiac muscle cells. And intercalated discs also provide the cell junction between a pacemaker cell and a contractile cell. Intercalated discs are made of two things. One of those things is desmosomes, and that's going to help prevent the muscle cells from pulling apart during contraction. The other of those things is gap junctions.
Gap junctions. Aha!
So that's why intercalated discs matter so much, and we keep making you look at them, even though students complain, oh, they're hard to see, why do I need this? Gap junctions are critical to the whole thing I just drew. If you didn't have gap junctions, none of this would work.
Because that's what allows the neighboring depolarization of other cells, right, to send the impulse through.
Exactly. So that's how the pacemaker cells are able to excite the contractile cells, and that's how one patch of contractile cells is able to excite the neighboring patch. This only works because of gap junctions.
Putting this together with the two charts there makes a really, really big difference.
Good, I'm glad.
Okay.
Because for this particular set of processes, there's a lot of steps, but the order of the steps is really important.
Okay, so since you've got this up, so in that first step, the SA node is firing, so that one's depolarizing, right?
Yeah, that's what we mean when we say firing. It's basically shorthand for depolarizing.
Okay, and then, so in that second step, is that the atria depolarizing as well?
The second step is just any contractile cell that has been depolarized as a result of the pacemaker potential.
Okay. And then you get into, so in the steps that you have, what about the beginning of the ventricular depolarization?
Same thing. So what part of the nodal system would cause ventricular depolarization?
The sodium.
Not quite. So you said the words of it earlier. After the impulse travels down the bundle branches, where does it go?
To the Purkinje fibers.
So bundle of His or AV bundle, the bundle branches, and the Purkinje fibers, those are all also doing this. They're just doing it after the SA node does. And then they're going to excite the neighboring contractile cells, which produces ventricular contraction.
Gotcha.
Alrighty, so I think I'm going to stop the recording now since we've hit a lull.